Help in Pharmaceuticals in association with Higher Secondary Principals Forum.
Hello students, we meet again for the second chapter in biology, sexual reproduction in flowering plants. In this chapter, students, we are going to be discussing about sexual reproduction in angiosperms. So we are not going to be discussing about any other mode of reproduction. We are only going to be going in details with sexual reproduction in flowering plants or angiosperms. So what is a flower? A flower is a very fascinating organ in angiosperms. A flower is also considered to be of significance in human life. Why? Because a flower has social value, a flower has aesthetic value, a flower has ornamental value, religious and cultural value. Now, this is with regards to human life. But what about the plants or the angiosperms? The flower is considered to be a very fascinating reproductive structure or organ in them. Why? Because it is through this flower that reproduction, that is sexual reproduction, takes place. So let us try to understand a little more about the longitudinal section of a flower. And it is said to be having 4 whorls. Now why do I say 4 whorls? It's because this particular flower is a bisexual flower. In the previous class, we have seen what do you mean by bisexuality. So if you notice here, this is a bisexual flower having 4 whorls. The outer accessory whorls, that is calyx made of sepals, corolla made of petals. So as you can see over here, this is the outermost whorl made up of sepals. The next whorl inner to the calyx is corolla, which is made up of your petals. The inner reproductive whorls are enrichem, which is made up of stamens. These structures are your stamens. And gynecium, which is made up of pistil or carpel. So you can see over here, this is the carpel or your pistil, which is made up of your ovary, style, and the sticky stigma. So this is a typical flower, which is having 4 whorls, which means it is a bisexual flower. Now, in sexual reproduction, we are going to be highlighting upon the various events which lead to sexual reproduction in flowering plants. So we have pre-fertilization events, fertilization event, and post-fertilization events. In pre-fertilization events, we are going to be discussing 2 processes. One is microsporogenesis and the other one is called megasporogenesis. Now, what do you mean by microsporogenesis and megasporogenesis? Please bear in mind, students, in my previous class, I have already discussed with you, with regards to sexual reproduction, we need to form the gametes. In flowering plants, the gamete here, or the male gametophyte, should be the pollen grain. So we need to form the pollen grain. And to form the pollen grain, we require a process that is known as microsporogenesis. Likewise, you require the egg. Now, the egg is formed inside a structure known as the embryo sac, or it's also known as the female gametophyte, through a process which is called as megasporogenesis. Once the microspores and megaspore is formed, that is the pollen grain and the egg is formed, it will lead to the next event, which is called as the fertilization event or syngamy. Now, in angiosperms, students, please bear in mind that this fertilization is of a different type. It is a characteristic feature which is exclusively found only in angiosperms, and it is the type of fertilization which is called as double fertilization. We are going to be discussing it in details later. And of course, you have the post-fertilization events, which include endosperm formation, embryo development, seed and fruit development. So, to understand about microsporogenesis, let us try to understand the parts of the flower, which include stamen, microsporangium and the pollen grain. Let us begin with the stamen. The stamen is also known as the microsporophyll or the male reproductive organ in a flower, or the male reproductive structure in a flower. So what you see over here is a single stamen which has two parts to it. It has the filament and it has the anther. So this particular structure that you have over here, the anther, as you can see, is bilobed. It has two lobes. That's why it is called as a bilobed anther. Okay? Now, inside the bilobed anther, what do you have? To see that, it's a cross section that is shown to you in this figure. In this figure, you can see this is the bilobed anther. This is one lobe. This is the other lobe. And inside each lobe, there are two theca. Now, what are these theca? Sac-like structures, as you can see. This is one, this is two. In each lobe, as you can see, there are two theca. They are known also as the pollen sac, also known as the microsporangia. Plural, singular would be microsporangium. So how many microsporangia are there inside this bilobed anther? There are 4 microsporangia, also known as 4 pollen sacs. Moving on, a little more in details about the anther structure. As you can see here, the magnified view of the same image that you saw in the previous slide. The anther is bilobed. It is dithecous. Why is it called dithecous? Because each lobe has 2 theca. Di refers to 2. So since each lobe has 2 theca, it is a dithecous anther. And you can see line of dehiscence. This is the line of dehiscence that is shown to you. This is the longitudinal line of dehiscence, and this is the other one over here. This is the line of dehiscence. What do you mean by line of dehiscence? When this structure, that is the anther, gets matured, the pollen grains that are formed inside, which are matured, also have to be dispersed. So for that purpose, you will see that there is going to be a split that is going to be noticed longitudinally along this line, and it will cause the anther to split open, causing the release of the pollen grains which are formed inside it. That's why this line is known as the line of dehiscence. And of course, you have 4 microsporangia or 4 pollen sacs inside this bilobed dithecous anther. Let us move on to understanding the anther wall. Now, if you look over here, this is your bilobed anther having the dithecous condition or the 4 pollen sacs. Each pollen sac let us focus on, and you will be able to see that each pollen sac is surrounded by several layers. That's why they are called as your wall layers. Now, what are these wall layers? Let us understand these wall layers. So the outermost layer of this particular microsporangium or the pollen sac is your epidermis, this one. The single layer made up of the single epidermal cells is your epidermis. Inner to the epidermis is again another single layer which is known as your endothecium. The green layer inner to the epidermis is your endothecium. Again, it is a single layer of cells inner to epidermis. Inside the endothecium, you have 2 to 3 middle layers. Inner to the endothecium, you have 2 or 3 rows of middle layers. Now, what are these middle layer cells going to be doing? These are nutritive in function, so they are going to be nourishing some cells called as the microspore mother cells and the microspores. We'll understand that a little later. Inner to the middle layers, middle layer, what did I say? These are nutritive cells which nourish the microspore mother cells and the microspores that are getting formed inside the pollen sac. Inner to the middle layer, we have the tapetal layer or the tapetum. Now, this tapetum is again a nutritive tissue which is going to be nourishing the developing pollen grains or the pollen. And inside the tapetum or inner to the tapetum, we are going to be having this tissue which is known as the sporogenous tissue. So as you can see over here, these are your middle layers. Inner to the middle layer is your tapetum. This one, the yellow colored layer that you can see is the tapetal layer, and inner to the tapetum is going to be this structure, which is a group of cells known as your sporogenous tissue made up of sporogenous cells. So the layer which is in immediate contact with the sporogenous tissue is the tapetal layer or the tapetum. And why is it so? It's because this is the tissue which is ultimately going to be nourishing the pollen grains that are getting formed in the sporogenous tissue. So I hope the anther wall layers are clear to you. Moving on. You can see over here, this is again a highlighted view of the anther wall, and this diagram is there in your textbook. So outermost layer is your epidermis, inner to the epidermis is the endothecium, then you have the middle layers followed by the tapetal layer and of course your sporogenous tissue made up of your sporogenous cells. Now, before I move on to making you understand what do you mean by microsporogenesis, I want you to focus on the sporogenous tissue. What is a tissue, students? A tissue is a group of cells which perform a similar function. So, what do you understand by a sporogenous tissue? It is a tissue that is ultimately going to be giving rise to spores. In other words, the microspore or the pollen grain. Now, these sporogenous tissue that you have made up of sporogenous cells are diploid cells. Diploid meaning each cell of this tissue is having paired set, that means paired set of chromosomes, right? Okay, so that's why it is called as a diploid cell. So, the sporogenous tissue made up of your sporogenous cells are diploid cells, and they are the precursors that will give rise to your pollen grains. So what happens here now in microsporogenesis, that is the formation of the microspore inside the anther, is that the sporogenous cells get transformed to behave as microspore mother cells. What happens here? The sporogenous cells, they transform themselves to behave as the microspore mother cells. Again, these microspore mother cells are diploid cells which are going to be giving rise to the microspores. And how does it take place? With which cell division? Microsporogenesis takes place with the help of meiosis or reductional division. As you can see here, microsporogenesis, let's focus on this. This is the sporogenous tissue, say for example, students, okay, which is made up of say around how many? 5 of your microspore mother cells or your pollen mother cells. Another word for microspore mother cells are your pollen mother cells. So from where have these pollen mother cells or microspore mother cells come? They have got transformed from the same sporogenous tissue itself, which is made up of your sporogenous cells. Keeping that in mind, let's move on forward. Now, these pollen mother cells, each of them have the ability to undergo meiosis or reductional division. So, each one now starts undergoing meiosis. So, meiosis is divided into meiosis 1 and meiosis 2. So, in meiosis 1, which is reductional division, you will notice that if each pollen mother cell undergoes first meiotic division, it will give rise to 2 daughter cells. And then the second meiotic division will yield 4 daughter cells. So, as you can see over here, what has happened? Each of the pollen mother cell has undergone meiosis 1, wherein each of the pollen mother cell has divided, only the nucleus has divided. As you can see, 2 nuclei in each of the cell. That means meiosis 1, karyokinesis is completed. Once meiosis, karyokinesis is completed, we move on to the next division, which is your meiotic 2 division. Meiotic 2 division is like your mitosis, which is equational type of a division, wherein you will notice from each pollen mother cell, we are going to be able to get 4 microspores, and they are going to be in the form of a tetrad. So what you see over here are your tetrads that are formed from the pollen mother cells. So you have 5 pollen mother cells. At the end of meiosis, you will get how many tetrads? 5 tetrads. Each tetrad is having 4 microspores that are formed through meiosis. And as I already mentioned to you earlier, meiosis is reductional division, and therefore these tetrads that are formed, which are carrying the 4 microspores, each of the microspore is going to be a haploid microspore. So what happens now to this tetrad? How is it that the 4 microspores are joined together to form a tetrad condition? It is because of a substance known as callose. Now, callose is a substance that is synthesized or secreted by the endothecium. Remember what is endothecium? Going back to my previous slide, this endothelial layer, here, you can see it over here. This is the endothecium. This is the endothelial layer which is responsible to produce the callose chemical, which helps to keep the 4 microspores united or together. It is like a cementing substance. So what happens next is, the moment the tetrads are formed, the microspores, they grow, they try to get developed further, and as a result you will notice the cementing substance between them that was joining them together gets dissolved. Now, how does that callose cementing substance get dissolved from each of the tetrads and makes the microspores to be free? It is because of an enzyme that dissolves the callose substance. That enzyme is called as callase. And callase is the enzyme that is secreted by the tapetum. Okay? So what you have now are the 4 free microspores. The callose cementing substance is dissolved with the help of the enzyme called callase enzyme. How did you get these 4 microspores? You got these 4 microspores from a single pollen mother cell. So, listen to me very carefully. If I have 4 pollen mother cells, how many microspores will I get at the end of meiosis? Can you answer this question, students? If I have 4 pollen mother cells, each pollen mother cell is a diploid cell. At the end of meiosis, in these 4 pollen mother cells, how many microspores will I get? I will get 16 microspores. Yes or no? Okay. So these 4 microspores now, what happens to them? They now get transformed into the matured pollen grain. Now, to transform into the matured pollen grain also, it is a process. That is the formation of the pollen grain. Once the microspores are formed, they have to undergo further development to get transformed into the pollen grain. Now, how does that take place? Because they have been fed upon by the tapetal layer and the endothelial medial layers, and as a result they grow, they develop and transform themselves into a matured pollen grain, which is able to bring about fertilization. So let us try to understand the development of the pollen grain now. Here you can see again, I have shown you a single pollen mother cell. This single pollen mother cell with the diploid nucleus undergoes meiosis 1 to form 2 cells, basically 2 nuclei inside the cell, followed by meiosis 2 having 4 nuclei. As a result, what will happen now? Cell walls are going to be laid around each of the nuclei to give rise to 4 microspores, which are joined together by the callose enzyme to form something known as the tetrad. The tetrad will get dissolved by the callase enzyme. Now each of this microspore now this is a microspore, will undergo development to become the matured pollen grain. Now how does that take place? Let's understand here. So what you see over here is a microspore. This microspore now undergoes transformation and becomes 2-celled. Now how does one cell become 2-celled? By what cell division here? It is mitosis. No more meiosis now, it is going to be mitosis division. So this single-celled microspore undergoes mitosis to become 2-celled. One is a large vegetative cell and the other one is going to be a small tiny generative cell. You can see here clearly, this is going to be the vegetative cell, and in the vegetative cell you are going to be having the vegetative cell nucleus. These are vacuole, students. Okay, this is the vegetative cell nucleus and the yellow portion is the vegetative cell cytoplasm. In the vegetative cell cytoplasm, you will be having another tiny cell which is the generative cell which floats in the cytoplasm of the vegetative cell. So you can see here, in the cytoplasm of the vegetative cell, you will have the generative cell with its nucleus which is spindle shaped. And this generative cell, a tiny cell, will be floating in the cytoplasm of the vegetative cell as seen in the figure. So finally, what do you see over here is going to be the generative cell with its nucleus, the vegetative cell with its nucleus and the vacuoles which are present. And later, you will also notice that this generative cell again undergoes division. What division? Mitosis. So what will happen now? This generative cell divides mitotically to produce two daughter cells. These two daughter cells now are going to be called as the two male gametes. So what are they going to be called? They are going to be called as the two male gametes. So we have two things to keep in mind here. One is that this is a pollen grain which is already matured, and this is also a pollen grain which is also already matured. So pollen grains which are matured can either be in two-celled condition or three-celled condition. So what do you understand by a pollen grain which is two-celled? It is having the vegetative cell and the small generative cell. What will be a three-celled pollen grain? A three-celled pollen grain would be a structure wherein you will notice there will be the vegetative cell and two male gametes. How have you got the two male gametes? You have got them through mitosis of which cell? The generative cell. So you have either a two-celled pollen grain or a three-celled pollen grain. As I already mentioned to you, a pollen grain is made up of a large vegetative cell with its nucleus, a small generative cell with its spindle-shaped nucleus, floating in the cytoplasm of the vegetative cell. And in over 60% of the angiosperms, students, bear in mind, pollen are shared in two-celled stage. What is a two-celled stage? This is the two-celled stage with the vegetative cell and the generative cell. So in over 60% of the angiosperms, the pollen grains, they are released from the anthers in their two-celled condition. Whereas 40% of the angiosperms, pollen grains are going to be released in what stage? The three-celled stage, that is this one, with the two male gametes. Clear? Moving on, let us understand the outer walls of the matured pollen grain. So the outer layer is called as exine. This is the outer layer which is called as the exine, which is made up of a substance known as sporopollenin. Coming to that a little later. The inner layer is called intine. It is made up of cellulose and pectin. So this is the inner layer and this is the outer layer. Now this outer layer could be smooth or rough. Now outer layer is made up of a substance called sporopollenin. To tell you a little more about sporopollenin. Sporopollenin, students, is a highly chemically inert or stable biological polymer. It is highly resistant also to a lot of, it is resistant to high temperatures, strong acids, strong alkalis, etc. So it is a very stable or an inert biological polymer which is made up of carbon, hydrogen, oxygen, and it does not get affected by any kind of pressures or temperatures in the environment, or even for that matter strong acids and strong alkalis. And it is seen on the outer wall of the pollen grain, which is known as your exine. The inner layer is called intine. And this inner layer, which is called intine, is responsible also to form the pollen tube. Now, pollen tubes are structures which will be carrying the two male gametes for fertilization. So, who forms the pollen tube? It is with the help of the layer which is called as intine, which is the inner layer, inner to the exine. And I also want to draw your attention to something called as a germ pores. Now, what are these germ pores? These are openings. As you can see here, there is a single germ pore for this pollen grain. Here also there is a single germ pore. Now, these germ pores are openings or pores through which the pollen tubes will be emerging outside. See, each pollen grain is a continuous membrane. There are no openings. So you require the tube, the pollen tube, to emerge outside, and that is only going to be possible with the help of germ pores. And there would be about 2 or 3 germ pores for every pollen grain. The structure of the pollen grain is clear. We have seen the external structure. We have also seen the internal structure of a matured pollen grain. Moving on to now some advantages and disadvantages of these pollen grains. Now, pollen grains are known to cause pollen allergy. They cause severe allergies, example, parthenium or carrot grass, it's also known as. It's a weed which is responsible to cause pollen allergy in humans. You also have some kind of other ailments called bronchial afflictions that are associated with the respiratory system because of these pollens that are released in the air or environment. Pollen release or pollen allergy can also lead to chronic respiratory disorders like asthma, bronchitis, etc. What are the advantages of pollen? We have something called as bee pollen, and this bee pollen is a mixture of the nectar, saliva of the bee and the pollen put together. Now, bee pollen is also used as food supplement for the reasons already mentioned, that it is rich in vitamins, minerals and antioxidants. It is also known to increase the performance of athletes and race horses. Now, have a look at this point. It is preserved as microfossils also. You must have heard about fossils. If you go to any museum, students, you will get to see the organisms, be it plants or animals, and their parts which are preserved and kept. You can preserve the whole animal or the plant, or parts of the animal or the plant as well. So here we have something called as microfossils. Microfossils are fossils which are not visible to the naked eye. You need to take the aid of a microscope to view them. That's why they are called as microfossils. So pollen grains are also called as microfossils. And fossil is something that is very old, age long, right? It is something that is years or millions of years old, and it is still intact, which means pollen grains can also remain intact for several thousands of years. When I say they can remain intact, doesn't mean that they are viable. They may not be viable to cause fertilization or bring about fertilization. But what I'm trying to tell you is the external structure of the pollen grain can remain intact. And that's why they are preserved as microfossils. How is that possible? How do you think that these pollen grains can be preserved as microfossils for thousands of years and the external structure will be stable? It will not change. It's all because of this substance known as sporopollenin, which I already told you, it is a very stable biological polymer and it does not get affected by temperatures, pressure, alkalis, acids, etc. Now, we have finished discussing about the stamen. Let's move on to understanding the pistil or the carpel. Now, as you can see over here, this is a single carpel or a pistil which is made up of the ovary, the style and the stigma. These are the three important parts of a pistil or a carpel. Again repeating, you have the ovary, inside which you will have the ovules, the style which is the stalk, and the stigma which is highly receptive for the pollen grains. Now, moving on to the type of pistil or carpel. Here we have monocarpous condition and multicarpous. What do you mean by monocarpous and multicarpous condition? Monocarpous is a condition where you will see that there is going to be a single ovary, style and stigma in a flower. That is a monocarpous condition. What do you mean by multicarpous or multicarpillary condition? Multicarpillary would mean that there are many carpels, more than one, isn't it? That's why we use the term multicarpillary. So multicarpillary would mean where you have more than one carpel fused together or together. So you could either have multicarpillary apocarpous condition or multicarpillary syncarpous condition. Multicarpillary apocarpous condition meaning, as you can see over here, how many carpels are shown to you in this figure? You can see 1, 2, and this is the third one on the other side. But are they joined together on the thalamus of the flower here? No. They are no doubt 3 in number, but they are free. They are not fused together. Therefore, this condition is going to be called as apocarpous condition. What do you understand by apocarpous condition? It means that there are more than one carpel and they are free and not fused. So I can call this condition as a tricarpillary apocarpous condition. Tricarpillary apocarpous. Why? Because there are how many carpels? 3 carpels. That's why tricarpillary apocarpous. Why? Because there are 3 carpels which are free and not fused. Now look at this one over here. Now what do you see over here? How do you know how many carpels are there? I can clearly see that there are 3 carpels. This is the first one, this is the second one, and on the other side, this is the third one. But what do you notice? Compare this figure to this one, and what do you observe? If you observe this figure and compare it to the previous one, you will notice that the carpels, there are 3 in number and they are fused at what level? At the level of the ovary they are fused. As you can see, the style and the stigma may be free, but they are still fused at what level? At the ovary level. So what condition is this going to be called? I can call this condition as multicarpillary syncarpous condition. So you have this flower which is made up of the gynoecium, and that gynoecium is carrying carpels. How many in number? 3 in number. And are they free or fused? They are fused. So I can call this condition as multicarpillary syncarpous condition of the gynoecium. Over here also, if you notice, fused carpels, how many in number? 3 in number. So all these 3 put together also you can categorize them as syncarpous condition. Here, this is the diagram that you can see at the bottom, which also tells you that these are fused carpels and these are free carpels at the level of the ovary and the ovule. Students, this diagram is shown to you. All right, moving on. In understanding the structure of the ovule now. Now, please bear in mind, students, pistil or carpel, okay, is the one that is going to be carrying the megasporangium. Now what is the megasporangium? Megasporangium is the ovule. So megasporangium is the ovule, and what is the microsporangium? What we have earlier discussed, it is the pollen sac. So try to relate to what I am trying to draw your attention to. If the microsporangium is the pollen sac, what is the megasporangium in the female part of the flower? It is your ovule. Getting me? And what does the ovule carry or contain inside it? It will be carrying the egg or the embryo sac. Like how we saw in each microsporangium or the pollen sac, we had the pollen grains that are getting formed. In the same manner, inside the ovule, we are also going to be creating or forming something which we call it as the embryo sac, which contains the egg inside it. So the megasporangium or the ovule, what you see over here are 2 types of ovules shown on the slide. One is called as the orthotropous ovule, the other one is called as the anatropous ovule. Now what is the meaning of an orthotropous ovule? It is a straight ovule. You can see this is the body of the ovule and this is the stalk or the filament of the ovule. The same thing now if you invert it. Invert the body, okay, by 180 degrees. So if you invert the body of the ovule by 180 degrees, what will happen? The body will bend 180 degrees, and as a result, the straight ovule or the orthotropous ovule becomes what ovule? Anatropous or inverted ovule. So if you invert the body of the ovule by 180 degrees, you get another ovule, which we call it as the anatropous ovule. In your textbook, students, you have this figure. This is the figure that is given to you in your textbook. What type of an ovule is this? In your textbook, it is highlighted as the anatropous ovule. But in reality, anatropous ovule is what this diagram looks like. And in the textbook, also, what is projected is an anatropous ovule, but it is shown to you in the reverse direction. So let us try to understand the structure of a typical anatropous ovule now. So what you see over here is an anatropous ovule having the stalk, which is called as the funiculus. The funiculus and the body of the ovule. The point at which the body of the ovule meets the funiculus is called as hilum. So hilum is the point or the junction where the body of the ovule meets the funiculus or the stalk of the ovule. Now you may wonder, where is the stalk going? The stalk is attached to the tissue which is called as the placenta. And placenta is a tissue seen on the inner wall of the ovary. Where is the ovule found, students? This is the ovule, for example, which is found inside the ovary, and the stalk, this is the stalk which is attached to the inner wall of the ovary with the help of placental tissue. Moving on now. So we saw what is funiculus, we saw what is hilum. Let us try to understand the structure that is the body of the ovule. Starting from the outside, we have two layers here, which is called the outer integument and the inner integument. Outer integument and inner integument. What are these? These are protective sheets or protective layers for the ovule structure. So inner to the outer integument and inner integument, you have this tissue. What is this tissue known as? This tissue is called as the nucellus. And this tissue is made up of cells called as nucellar cells. So what is the nucellus? Nucellus is the tissue inside the body of the ovule. And what is the nucellus made up of? It is made up of cells called as the nucellar cells, which are diploid cells. Now, out of these nucellar cells in the nucellus, one nucellar cell is selected. Only one nucellar cell is selected from thousands of nucellar cells, and this selected nucellar cell is the one that will begin the process of megasporogenesis. So what is megasporogenesis? Megasporogenesis is the formation of the megaspore in the ovule. Formation of the megaspore inside the ovule with the help of the single selected nucellar cell. So that single selected nucellar cell now starts behaving as the megaspore mother cell, MMC. It's a diploid cell which is now going to begin with megasporogenesis. So you can see over here, let's follow it. You have the placental tissue attached on the inner wall of the ovary, to which you have the ovule that is attached to. Now what kind of an ovule do you see in this figure? It is an orthotropous ovule or a straight ovule that I have drawn. Now let's move on further. This is the nucellar tissue or the nucellus, in which one nucellar cell is selected which behaves as the mother cell or the megaspore mother cell. This selected megaspore mother cell, which is a diploid cell now, begins with meiosis 1 and meiosis 2 gets completed, resulting in the formation of how many megaspores now at the end of meiosis? 4 megaspores. The same repetition, students. One single microspore mother cell or the pollen mother cell undergoes meiosis to give rise to how many microspores? 4 microspores. In the same manner, this mother cell or the megaspore mother cell undergoes meiosis to give rise to how many megaspores? 4 megaspores. Now, out of these 4 megaspores that you see in the nucellar tissue, only 1 persists. The other 3 will degenerate. As you can see over here, what has happened? There is only 1 functional megaspore that grows further. The other 3 become insignificant, they shrink and they get absorbed. That means they die off. So what remains out of the 4 megaspores is going to be a single functional megaspore that later on goes on to become the embryo sac or the female gametophyte. What is the male gametophyte? Pollen grain. What is the female gametophyte? It is the embryo sac. And this embryo sac is formed how? It is formed from a single functional megaspore. And this development of the embryo sac is called monosporic development. Why is it called monosporic? Because the embryo sac gets formed from a single megaspore. That's why the development is called as monosporic development. So you see here, this is the functional megaspore. The 3 will degenerate in the nucellar tissue itself. And what will happen? You will notice that this functional megaspore starts growing inside the ovule. This megaspore that starts growing now, how does it grow? It grows by cell division. What kind of cell division? Mitosis. So this single megaspore, is it haploid or diploid, students? How has it been formed? Through meiosis. And therefore, this particular megaspore is going to be a haploid megaspore, which is how many celled? It is just a single cell with its single haploid nucleus. Where is it present at the moment? It's present inside the nucellus. And nucellus is a part of the ovule, right? Okay. Now what will happen to this megaspore? The megaspore has to transform itself into the female gametophyte, also known as the embryo sac. So what happens is, this particular megaspore undergoes mitotic divisions. But what type of mitotic divisions? Karyokinesis only, which means only the nucleus starts dividing and not the cytoplasm at first. So you notice here, 1 cell nucleus becomes 2 nuclei inside the megaspore. 2 will become 4. And what will happen to the 4? 2 nuclei will move at one end of the megaspore, and the other 2 will move at the other end of the megaspore. Then 4 will become how many? The next cell division, it will become 8. So you will have 1, 2, 3, 4, 5, 6, 7, 8. You can see over here, 1, 2, 3, 4, and here you have 5, 6, 7, and 8 nuclei, right? So 8 nuclei have been formed by how many mitotic divisions? 3 mitotic divisions. So the first mitotic division gave rise to 2 nuclei, the second mitotic division gave rise to 4 nuclei, and the third mitotic division will now give rise to the 8 nuclei. Now have a look at this. What happens to the nuclei? The nuclei start rearranging themselves. Follow the diagram. The 8 nuclei start rearranging themselves, and you can see that 3 nuclei remain at one end, 3 nuclei remain at the opposite end, and 2 nuclei come in the center of this structure now. So what will happen now? You notice in the next figure, cell walls start getting laid. So karyokinesis is over. Now we begin with cytokinesis. What is cytokinesis? Cell walls are going to be laid for the nuclei. So 3 cells at one end, 3 cells at the opposite end, and 2 cells in the center. So what you get right now is a structure which is called as the matured embryo sac or the female gametophyte, which is again very characteristic feature of these angiosperms, because it is 7-celled, 8-nucleated. Isn't it so? Why is it 7-celled, 8-nucleated? Have a look at this. 3 cells on top, 3 cells at the bottom. Total 6 cells. And 1 cell, this is the central cell. It is carrying how many nuclei inside it? 2 nuclei inside it. So total how many cells? 7 cells, 8 nuclei. So this is the characteristic feature of the embryo sac or the female gametophyte. It is 7-celled, 8 nucleated. Did you understand the structure of the embryo sac, students? So let's go through the structure of the embryo sac now. We have at one end the 3 antipodals, which are haploid cells. And what is their role now in the embryo sac? They are going to be nourishing the developing embryo. At the opposite end, you have the egg apparatus, which is made up of the 2 cells, which are called synagids, and in the center there is one cell called as the egg cell. So the egg cell along with the 2 synagids are known as the egg apparatus. In the center we have the central cell with 2 nuclei. The 2 nuclei are again haploid nuclei. So we have the haploid egg apparatus cells, the haploid antipodals, and the central cell, which I will not call diploid. Why? Because the 2 nuclei are not fused. So rather I would use the term N plus N in this case. So this is what you see is a typical structure of the embryo sac. In this unit now, what did we discuss? We discussed megasporogenesis, which is the formation of the megaspore. Once the megaspore gets formed, the functional megaspore, okay, this functional megaspore then gets transformed into the embryo sac. This is what is the embryo sac that you have. How did the transformation of the single megaspore into the embryo sac take place? Through the division which is called as mitosis. So, mitotic divisions first with the help of karyokinesis, followed by cytokinesis, led to the formation of the typical embryo sac or the female gametophyte having antipodal cells, the egg apparatus, and the central cell with the 2 polar nuclei. Now what happens next is going to be the fertilization event, wherein this egg cell which is there is going to be fertilized by one of the male gamete. Remember, the pollen grains are shared either in their 2-celled condition or their 3-celled condition. In 3-celled condition, you have the vegetative cell and the 2 male gametes. So one of the male gamete would go and fertilize with the egg cell. N and N fuse together to restore the diploid condition, which forms the zygote. And what happens to the other male gamete? The other male gamete goes and fuses with the 2 polar nuclei in the central cell to form the primary endosperm nucleus. And primary endosperm nucleus goes on to later become the endosperm or the nutritive tissue, which will nourish the developing embryo. From where is the embryo going to arise? From the zygote, which is formed by the fertilization. So to summarize today's class, we discussed about the flower, which is the reproductive organ in plants or angiosperms. We also saw the structure of the endometrium and the gynoecium, the stamen, the carpel or the pistil. We moved on to understanding what is microsporogenesis and megasporogenesis. We saw how a pollen grain gets formed. We also saw the formation of the embryo sac or the female gametophyte. In the next class, we are going to be discussing about double fertilization, pollination, and post-fertilization events, along with the discussion of some of the questions that have been asked for your board exam. Thank you, dear students, and have a nice day.
Prudent Scholars, powered by Lupin Pharmaceuticals.